Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to tackle a practice problem set that relates to our lecture on embryogenesis and development. Let's go ahead and get started with problem number 1. Problem number 1 says, which of the following signaling molecules coordinate uterine contractions during childbirth? Now, during childbirth, uterine contractions are coordinated by two main signaling molecules: oxytocin, which is a hormone that's released from the posterior pituitary, and prostaglandins, which are lipid-based signaling molecules that are produced locally in the uterus. Oxytocin stimulates the smooth muscle of the uterus to contract, and prostaglandins amplify and sustain those contractions. And together they create the rhythmic uterine activity that's needed for labor and delivery. So that means the correct answer here is D. Problem 2 says, which of the following associations of a primary germ layer and an adult organ is correct? A says endoderm, cardiac muscle. B says endoderm, lens of the eye. C says ectoderm, fingernails. And D says mesoderm, lining of digestive tract. The correct answer here is C, ectoderm, fingernails. The ectoderm is the outermost germ layer, and it's responsible for forming structures that interface with the external environment. So that includes the epidermis, which gives rise to the skin, hair, and nails, as well as specialized structures like the lens of the eye and the entire nervous system. Because fingernails develop from the epidermal layer, which comes from the ectoderm, this association is correct. The other choices are incorrect because they pair structures with the wrong germ layers. So cardiac muscle, like it's listed in choice A, this actually develops from the mesoderm, not the endoderm. The lens of the eye, this is derived from the ectoderm, not the endoderm. And the lining of the digestive tract, this comes from the endoderm, not the mesoderm. So only choice C correctly matches a germ layer to one of its true derivatives. Let's move on to problem 3. Problem 3 says, from which of the following layers does the notochord form? The notochord is a rod-like structure that plays a crucial signaling role during embryonic development, especially in the induction of the overlying ectoderm to form the neural tube. The structure itself arises from the mesoderm, which is the middle germ layer that's responsible for forming muscles, bones, the circulatory system, and supportive structures like the notochord. So therefore, the correct answer here is going to be B. As for the other options, the ectoderm, this gives rise to the nervous system and skin, not the notochord. The endoderm forms the internal linings like those of the digestive and respiratory tracts. And then the archenterion, this isn't even a germ layer at all. It's the primitive gut cavity that's formed during gastrulation. So while it's an important embryonic structure, it is not the source of the notochord. Only the mesoderm gives rise to the notochord. So once again, problem 3 is B. Problem 4 says, the influence of a specific group of cells on the differentiation of another group of cells is called blank. Induction refers to the process by which one group of cells influences the fate of another group of cells, often through signaling molecules or direct contact. Now, this is a foundational concept in embryology because it's how complex tissues and organs begin to form in a coordinated way. So, for example, during neurulation, the notochord, which is formed from the mesoderm, it releases signals that instruct the overlying ectoderm to become neural tissue. That's induction in action. It's not just about giving cells a push. It's about directing them towards a specific developmental path. So the correct answer here is D. Let's go over the other terms here. Competence, which is choice A. This refers to a cell's ability to respond to a signal, not the signal itself. Choice B, this is the process of aging or loss of cell division potential. Choice C, determination. This describes when a cell's fate is locked in, even if it hasn't visibly changed yet, but that fate was likely set by induction. So while related, none of these fit as directly as induction, which makes problem 4D. Problem 5 says, which of the following is likely to be found in the blood of a person who's pregnant? A says immunoglobulins produced by the fetus. B says fetal hemoglobin released from fetal red blood cells. C says progesterone produced by placental cells. And D says carbon dioxide exhaled from fetal lungs. Now, during pregnancy, one of the key roles of the placenta is to act as an endocrine organ. So it secretes several hormones to support and maintain the pregnancy. And one of the most important of these is progesterone. This hormone helps maintain the uterine lining, it prevents contractions, and it also supports the immune environment so that the fetus isn't rejected. And most importantly, because it's secreted by placental cells, progesterone enters the mother's bloodstream where it can be detected. So the correct answer for problem 5 here is going to be C. Let's still discuss why the other options are incorrect. So A mentions immunoglobulins produced by the fetus. But those don't typically enter maternal blood. In fact, it's the opposite. The mother's antibodies cross into the fetus for immune protection. Then choice B says fetal hemoglobin released from fetal red blood cells. But under normal conditions, the fetal and maternal blood supplies, they remain separate. So there's no significant mixing unless there's trauma or some sort of complication. That's why B is incorrect. D refers to carbon dioxide that's exhaled from the fetal lungs, but fetal lungs don't function for gas exchange before birth. Instead, the placenta handles all of the oxygen and all of the carbon dioxide exchange. So again, the only reasonable answer here is C, because it is made by the placenta and it circulates in the blood of the pregnant individual. 5 is C. Let's move on to problem 6. 6 says a cell releases a substance that diffuses through the environment, resulting in differentiation of a nearby cell. This is an example of what type of cell-to-cell communication. Let's walk through this one carefully. When a cell releases a substance that diffuses through the environment and affects a nearby cell, especially to influence something like differentiation, we're describing local signaling. And the key word here is nearby. That tells us that the signal doesn't travel far and it doesn't enter systemic circulation. This is exactly what defines paracrine signaling. In paracrine communication, a cell secretes molecules like growth factors, and these molecules act on neighboring signals. And this is really important in development where certain cells influence the fate of surrounding cells by secreting morphogens or differentiation signals that diffuse only short distances. So the correct answer here is going to be C. As a friendly reminder, autocrine signaling refers to a situation where the cell responds to the very substance it secretes, and that's not what we're seeing here. The signal is clearly affecting another cell. Juxtacrine signaling requires direct contact between cells, so no diffusion through the environment. And endocrine signaling involves long distance communication, where signals like hormones travel through the bloodstream to reach distant targets. That's way too broad in range for what's described here. So again, the best fit based on local diffusion and influence on a neighboring cell is paracrine signaling. 6 is C. Problem 7 says, a cancer cell is removed from a patient and cultured. The cells in this culture seem to be able to divide indefinitely with no cellular senescence. Which protein is likely activated in these cells that accounts for this characteristic? So to answer this question, we need to recognize what it means when cells are described as dividing indefinitely without undergoing senescence. In most normal somatic cells, there is a natural limit to how many times they can divide. And this is because each time a cell divides, its telomeres, which are the protective caps on the ends of chromosomes, become shorter. Eventually, these telomeres get too short and the cell can no longer divide. However, cancer cells are notorious for bypassing this limit. And one way that they do this is by activating an enzyme called telomerase. Telomerase adds repetitive nucleotide sequences to the ends of chromosomes, essentially rebuilding the telomeres and allowing the cell to keep dividing without limit. This enzyme is typically inactive in most adult somatic cells, but it is highly active in many cancer cells, which contributes to their indefinite division. So the correct answer here is D. Moving on to problem 8. Problem 8 says, anencephaly is a rare physiological abnormality in which the cerebrum fails to develop. During which trimester of pregnancy would this disorder manifest? Now, to figure this out, we need to understand when the structures of the central nervous system begin forming during pregnancy. Anencephaly is a severe neural tube defect in which the cerebrum, the largest part of the brain, fails to develop properly. Now, the neural tube is the embryonic structure that eventually forms the brain and spinal cord, and this tube needs to close very early in development, typically by the 4th week of gestation. Because of how early this step occurs, any disruption in neural tube closure must happen in the first trimester, well before the fetus even begins to resemble a recognizable human form. Neural tube defects like anencephaly results from issues during this critical early window, often before a person even realizes they're pregnant. So the correct answer is the first trimester. Let's briefly go through the other options. The second trimester is when organ systems continue maturing and growth accelerates. But by this point, the neural tube has long since closed. The third semester involves rapid brain growth and development of cortical complexity. But again, the foundational brain structures are already in place by then. And any trimester is too general and incorrect because this particular abnormality is very specific to early embryonic development. So in short, anencephaly originates from a failure in a critical developmental event that occurs very early in pregnancy, making the first trimester the correct answer. Problem 9 says, which of the following is false with regard to adult stem cells? A says they retain inherent pluripotency if harvested from selected organs. B says they are less controversial than embryonic stem cells. C says they require treatment with various transcription factors. And D says there is a reduced risk of rejection if the patient's own stem cells are used. To answer this question, we need to know a fundamental limitation of adult stem cells, and that is that they are multipotent, not pluripotent. That means they can give rise to several types of cells, but only within a certain lineage. Pluripotency refers to the ability to become nearly any cell type in the body, which is a property of embryonic stem cells, not adult ones. So that means the false statement here is choice A. All 3 of the other statements, B, C, and D, those are correct. Let's move on to problem 10. 10 says a child is born with an imperforate anus in which the anal canal fails to form correctly and the rectum is not connected to the outside world. This pathology is most likely accounted for by a failure of blank. So in this question, we're looking at a developmental defect where a child is born with an imperforate anus. That means the anal canal didn't form properly and it isn't connected to the outside world. So we need to consider which developmental process, when disrupted, would lead to this structural abnormality. The correct answer is apoptosis, which is the programmed cell death that plays a critical role in shaping structures during embryogenesis. Apoptosis is not just about eliminating damaged cells. It's also essential for normal morphogenesis. For example, apoptosis is how we separate the digits in our hands and feet. Similarly, in the development of the anal canal, apoptosis clears tissue to open up the correct passageway to the outside. Without this process, the anal opening may remain blocked, and that's going to lead to the condition that's described in this problem. Now, that means the correct answer is C, but let's work through why the other choices don't fit as well. So A says cell differentiation. This refers to when cells take on specialized roles, like becoming epithelial cells or muscle cells. That's important for forming tissue types, but a problem here wouldn't directly cause a mechanical blockage of the anal opening. Choice B says cell determination. Now, this comes before differentiation. It's the point at which a cell becomes committed to a specific lineage. Again, this is important, but it doesn't directly explain the failed connection to the outside world. Finally, D says neurulation. This is the formation of the neural tube, which gives rise to the central nervous system. Now, that happens earlier in development, and it affects brain and spinal cord formation, not the lower GI tract. So putting this all together, the most likely explanation is going to be a failure of apoptosis. So 10 is C. Problem 11 says, following a myocardial infarction, the heart often heals by the creation of a scar by fibroblasts. This is an example of blank. So when the heart undergoes damage from a myocardial infarction, also known as a heart attack, the tissue that dies cannot be regenerated by new functional cardiac muscle cells. Instead, the body responds by forming a scar that's composed of collagen and that's produced by fibroblasts. This scar provides structural support, but it lacks the contractile ability of healthy heart muscle. Because the original function of the tissue is not restored, this process is known as incomplete regeneration. So the correct answer here is B. The heart is one of several organs in the body, along with the brain, that has very limited regenerative capacity. Instead of regenerating the original tissue, the body compensates by forming a non-functional scar. So again, 11 is B. Problem 12 says, neurofibromatosis type 1 is a disorder that causes formation of tumors in multiple nervous system structures as well as the skin. While all cells carry the same mutation on chromosome 17, selective transcription of the genome appears to cause the most significant tumor genesis in which of the following primary germ tissue layers. Now, it tells us that what it's affecting is the nervous system structure as well as the skin. That's our biggest hint here. Tumors that develop in tissues derived from the ectoderm are going to display this kind of behavior. The ectoderm gives rise to both the nervous system and the epidermis of the skin, both of which are the primary tissues affected in this disease. So the correct answer for 12 is A. Let's move on to problem 13. Problem 13 says, which of the following shows the correct order of early development milestones during embryogenesis? In early embryonic development, the first key milestone is the formation of the morula. This is a solid ball of cells that results from the rapid division of the fertilized egg. This is followed by the formation of the blastula, which is a hollow sphere of cells with a fluid-filled cavity called the blastocele. The next major step is gastrulation, where the blastula is reorganized into a gastrula containing 3 primary germ layers: ectoderm, mesoderm, and endoderm. These layers will give rise to all of the tissues and organs of the body. Therefore, the correct chronological order of these developmental milestones is morula, blastula, then gastrula. And the correct answer here is C. Problem 14 says, a woman who is pregnant accidentally is given a single dose of teratogenic drug late in the 3rd trimester. The baby is born 3 days later. Which of the following is the most likely outcome? A says complete failure of organ development and death of the fetus. B says partial failure of organ development with survival of the fetus. C says serious disfigurement of the fetus. And D says respiratory distress at birth, but no long-term effects. Now, the most critical period is going to be during the 1st trimester. Exposure to teratogens during this window can lead to major congenital malformations, including things like structural organ defects and some severe development disruptions. But by the 3rd trimester, most of the major organs have already developed. So at this stage, the fetus is really just undergoing growth and maturing rather than forming new organs. So a single dose of this drug late in pregnancy is much less likely to cause structural abnormalities. However, some systems, like the lungs and brain, they're still maturing, so they can be temporarily affected. So that's why the most likely outcome here is going to be choice D, respiratory distress at birth, but no long-term effects. With that, we can move into our last and final problem. Problem 15 says, which of the following statements regarding fetal circulation is false? Let's go through each answer choice. A says in the umbilical cord, there are more arteries than veins. This statement is true. In the umbilical cord, there are 2 umbilical arteries and only 1 umbilical vein. The arteries, they carry deoxygenated blood away from the fetus to the placenta, while the single vein brings oxygenated blood back from the placenta to the fetus. B says the foramen ovale is the only shunt that connects 2 chambers of the heart. This is also true. The foramen ovale is the only fetal shunt that directly connects 2 heart chambers. Specifically, it allows blood to flow from the right atrium to the left atrium, and it bypasses the non-functional fetal lungs. C says blood flow in the ductus arteriosus is from the aorta to the pulmonary artery. This statement is false, and it is our correct answer. The ductus arteriosus does not carry blood from the aorta to the pulmonary artery. Instead, it carries blood from the pulmonary artery to the aorta. This bypasses the lungs by diverting most of the right ventricular output directly into the systemic circulation. Now let's still work through answer choice D. D says the ductus venosus is the only shunt that bypasses the liver. This is true. The ductus venosus is the only shunt that allows oxygenated blood from the umbilical vein to bypass the liver and enter the inferior vena cava. So it essentially streamlines delivery of oxygen-rich blood to the heart and to the brain. So again, the only false statement here is C. I hope this helps. Good luck, happy studying, and have a beautiful, beautiful day, future doctors.